Now, follow your dreams if you are sure they know the way how to fulfill that. So it is just your right to see the dream that is being followed by how that the God knows and the God always helps. Now, the next is cytoskeleton. It is found in all bacterial cocci or the bacilli. It is just like the architecture of any house. It lies just under cytoplasmic membrane, make the basic architecture and the skeleton of the bacterial cell, so that influences the cell shape, because the basic gray structure of the house can tell you the shape of the house. So it do take part in the cell division. Here one can see the spherical bacteria, and this is the cytoskeleton of this bacteria, which is FTSZ. And in case of the bacilli, one can see these cytoskeleton, which are MREB and FLSZ. So these make the basic architecture of the cell. The Vibrio cholera bacteria that also have the skeleton, and that is of a bit different. So these do have the extra with MREB, FTSZ, crescentin. Here you can see that makes the basic structure of the Vibrio cholera bacteria. Now the nutrient storage or the granules or the inclusion. These are of the different types found in the cytoplasm. These are the source of stored nutrients and energy in case of the unwanted situation. So that is just being used if there is starvation occur inside the bacterial cell. So these are the energy reserve being used in the general starvation of the cell. They stain with certain special dyes which are capable of making prominent inside the cytoplasm. So these appear as a metachromatic granule. So stains red with a methylene blue dye instead of the blue. Examples are the volatin, a reserve of the high energy intracytoplasmic inclusion. Various types of the lipid and the crystals are also present in the form of the storage or the granules or the inclusion inside the cytoplasm of the cell. Here one can appreciate the different color of the granules that are being stained by the different special stain for the granules. Here again one can appreciate the blue, pink and other. That is just the different molecules and the granules which are being stained by the different special stain. Now the ribosomes. Ribosomes are composed of the RNA and the proteins. These are the protein factories of the bacteria, sites of the protein synthesis as in the human cell, found in the cytoplasm of the bacterial cell. But the different size and the chemical composition, they have got the diameter of about 18 to 20 nanometer. Bacterial ribosomes are 70S type in size, with 50S one component and 30S other component. So these are the subunits, 50S subunit plus 30S subunit. Human cell ribosomes are 80S in size with 60S plus 40S subunits. So the difference in the ribosome RNA and the protein make the basis of the selective action of the antibiotics that only inhibit the bacterial protein synthesis by acting on the ribosomes of the bacteria, but they do not attack the ribosomes of the human cells in which these bacteria are residing, because they are of 80S, and the antibiotic which we are going to fire that only attack the 70S which is being contained by the bacteria. Now the RNA types, these are the messenger RNA, and mRNA transcribed from DNA carries code for protein synthesis. So this is just the messenger that taking message from the DNA and giving to the protein factories. Then transfer RNA transfer RNA, that is small tRNA, one for the each amino acid that is being made to make the protein. Then ribosomal RNA, which is written as small r and capital RNA, that is the structure part of the ribosome. So all these collectively make and take part in the formation of protein. Here one can see there is the larger subunit of the ribosome and this is the smaller subunit of the ribosome. These just overlap and just embrace one another, and here they are. If you look at this bacterial, 70S, the 50 subunit and 30 subunit. When they overlap, the total size is 70S swag per unit. And in case of the human, it is the 80S, 60S subunit plus 40S subunit. But because of their overlapping, they give the size of 80S. Now, the next is plasmids. These are the extra chromosomal, double-stranded circular DNA molecules can integrate or merge into the bacterial chromosomes, and they are capable of replicating independently. So these are self-replicating and they can replicate and can give the daughters and their other components. Occur in both gram-positive and gram-negative bacteria, and plasmids carry genes for the following function and the structure which the bacteria do have and do, that is the antibiotic resistance R factor mediated by the many enzymes, that is through the plasmid which are present in the bacteria. Resistance to the heavy metals like mercury present in the antiseptic like merthiolate and mercurochrome and silver, that is also being transferred through the plasmid. It is mediated by a reductase enzyme. Resistance to the ultraviolet light mediated by the DNA repair enzymes and the pili fimbriae which mediate adherence of the bacteria to the epithelial cells or the host cell, that is also through the plasmid. Exotoxin including several enterotoxin, these are also being produced under the order of plasmids. So the continuing the plasmid, different types of the plasmid can exist in one cell. Transmissible plasmid, mobilizable, as the name is self-explanatory, so these can be transferred or mobilized. They do transfer from cell to cell by the conjugation or the sex pilus, which I am going to tell you when the division of the cell of the bacteria will come. Large molecular weight 40 to 100 million contains 12 genes, that is transmissible plasmid. And to synthesize sex pilus and enzymes required for transfer present in few 1 to 3 copies per cell. Then the other type is non-transmissible, cannot be transferred, do not contain transfer genes, and because they remain silent there, so no transfer gene. They are small in size, molecular weight 3 to 20 million, present in many and 10 to 60 copies per cell, but basically they are non-transmissible. Here one can see the plasmids, the mobile plasmid, and this is the non-mobile plasmid. This mobile plasmid just have a flagellum, which is the sex pilus, and this is the pili, which is called the sex pilus, and here you can see that it is joining. This is the donor, this is joining to the recipient bacterial cell, and here because of their joining they are going to transfer the lactosome or the genetic material to this cell, which is called transposome. So the donor is going to give some plasmid from this donor cell to the recipient cells, and the end result is the new donor is having that plasmid, and that is the other new donor, they are capable of transferring their plasmid to the other cell. Here one can see the one cell is going to transfer, here the other bacteria is going to transfer their genetic material from this plasmid to this bacteria. So the plasmid is transferred with carrying the genetic material. Now, other plasmid encoded products are bacteriocins, the toxic proteins produced by the certain bacteria under the control of the plasmid, which are lethal for the other bacteria. Two common mechanisms of the action of the bacteriocins are, these are the poisons being produced by the bacteria for the other bacteria to killing the other bacteria. Degradation of the bacterial cell membrane by producing pores in the membrane of the other cell on which they do attack. Degradation of the bacterial DNA by the DNA enzyme that they produce the plasmid and destroy the DNA of the other bacteria. So the bacteriocins produced by the bacteria are colicins made by the Escherichia coli and the piocins made by the Pseudomonas aeruginosa, and they are all capable of killing the other bacteria. Bacteriocins are useful in treating infection caused by the antibiotic resistance bacteria. So these are being used instead of the regular or routine antibiotic. These bacteriocins are more capable of killing those bacteria. Here one can see the colonizing peptide and killing peptide, signaling peptide. These are all made by the bacteria. So these are just the bacteriocins, and as you can see in the next slide, they are capable of disrupting the cell wall and the cell membrane, so making the pore and just destroying the cell in the way, so that every content of the cell, the cytoplasmic, these leak out and the cell is, bacterial cell is going to die. That is just all because of the bacteriocin action. Here you can see the bacteriocin being produced by the bacteria itself and capable of destroying the other bacteria. Even they can get inside the cell, causing the signaling mechanism. So these are having the bacteriocin signaling peptide and also the killing peptide. The transposons, these are the small mobile piece of the DNA that can move between the chromosomal DNA and plasmid DNA. So from one DNA center to the other DNA center, they carry genes for the antibiotic resistance, toxins and the enzyme. They are nicknamed jumping genes because of their ability to move. So they are able to move, so that's called jumping genes. They cause mutation in the gene, so insert or alter the expression of the nearby genes which are present at one part of the DNA. So these are being inserted to change the function of that gene which is already there on that part of the DNA. They are not capable of self-replication. Several transposons can be located in the DNA, and here one can see the DNA, and here is the transposon. This transposon carrying the few genes that is going to jump and being inserted to the other part of the DNA which is continued here. So this is going to have the interruption and interrupted DNA sequence which is being inserted here, and this is the transposon. Here again one can see the different fragments of the DNA that can detach and that can be inserted to the plasmid. And here again these are the transposons in the pieces of the DNA which are being inserted to the DNA and regular and to the plasmid, which are the also the pieces of DNA.